All right, good afternoon, everyone. Welcome to today's webinar. To begin this webinar, my name is Erica Chow, and I'll provide a brief overview of the Bioenergy Technologies Office, or BETO, followed by presentations of state-of-the-art research by Dr. Jianping Yu of the National Renewable Energy Laboratory, NREL, and Douglas Elliott of Pacific Northwest National Laboratory, or PNNL. These two projects both won R&D 100 awards this last year for breakthroughs in bioenergy advances. Some housekeeping. Please record any questions and comments you may have during the webinar and send them to eere_bioenergy@ee.doe.gov. As a follow-up to the webinar, the presenters will provide responses to selected questions. Slides from this webinar will be posted online at this address here. And for any general questions regarding BETO, please email the same address listed here. As some additional background to today's webinar, I'd like to remind everyone that this is part of BETO's ongoing webinar series. Our webinars cover many of the office's activities and feature hot topic discussions that are relevant to the development of renewable fuels, power, and products from biomass resources. And you can find more information on our website in the events and webinar section. When it comes to oil consumption in our transportation sector, the challenges are pretty well known. Even as oil production increases in the US and prices have fallen recently, we still spend tens of billions of dollars on oil imports every year, and the health of our economy continues to be at the mercy of events in parts of the world that are becoming increasingly unstable. I think many of us recognize that in spite of where oil prices are today, we are likely to see upward pressure on oil prices over the long term, given that, of course, it is a finite resource. And regardless of the price, we all know that consuming petroleum and petroleum-derived products is producing carbon emissions, which is impacting the health of our children and our planet. Instead, we could be utilizing enormous resources that we have right here in the United States, reducing our carbon emissions and revitalizing rural America and creating jobs across many sectors of our economy. Two of our most important reports on the subject, the 2005 billion-ton study and the 2011 update to the billion-ton, projects that by 2030, the United States could sustainably produce more than 1 billion tons of biomass without competing with the food, feed, and fiber supply. That's enough to displace as much as 30% of petroleum consumed in the United States, and that doesn't even include the potential for agro-biofuels. And the office is currently working on the update to the billion-ton study that will be released this summer, so keep an eye out for that. BETO's mission is to accelerate commercialization of advanced bioenergy through R&D supported by public-private partnerships. To achieve its mission, BETO works to promote sustainable nationwide production of infrastructure-compatible biofuels through funding and outreach. Specifically, BETO's portfolio focuses on validating at least one pathway for a $3 gallons gasoline equivalent hydrocarbon biofuel with a 50% or greater reduction in greenhouse gas emissions compared to petroleum-based fuels by 2017. So BETO's mission is to support the research and development and demonstration of advanced technologies, bioenergy technologies. Our office spans the entire bioenergy supply chain. So feedstock supply efforts focus on RD&D to develop cost-effective integrated logistics systems. So this includes growing, harvesting, collecting, storing, pre-processing, handling, and transporting of quality feedstocks to biorefineries. Our conversion R&D program is focused on developing commercially viable technologies to convert terrestrial and algal feedstocks into liquid fuels, as well as bioproducts and biopower. Biochemical conversion R&D focuses on pathways for producing sugars and other carbohydrate intermediates, followed by conversion to finished fuels. And the thermochemical conversion R&D is focused on pathways producing bio-oil and gaseous intermediates from biomass, followed by upgrading to finished fuels. And then the another category we have is the demonstration and market transformation program that is focused on demonstrating and validating biomass conversion technologies through successful construction and operation of cost-shared pilot and demonstration scale integrated biorefineries. We also have cross-cutting programs in sustainability and strategic analysis, which proactively addresses sustainability of advanced bioenergy to enhance its benefits, public acceptance, and long-term viability. This program area conducts key analyses to guide planning and portfolio management and provides analytical basis for the R&D prioritization of target development and assessment of progress towards goals. And with that, we're on to our first speaker, Dr. Jianping Yu is currently a senior scientist and section supervisor of the Bioscience Center at the National Renewable Energy Laboratory. He has a Bachelor of Science in Biology from Sichuan University, a Master's in Science and Genetics from Chinese Academy of Sciences, and a PhD in Plant Biology from Michigan State University. And as you'll see, he seems to have taken the slogan, "Go Green," to heart. He has worked at NREL since 2005, mainly on cyanobacterial production of fuels and chemicals. And take it away, Dr. Yu.
Thank you, Erica, for the introduction. And welcome, everybody. So the project I'm going to talk about now is Cyanobacteria bioethylene. And we are fortunate that we received the R&D 100 award and R&D Journal Editor's Choice Award. So a trophy for the second award is shown here. Next, please. So the goal of this project is to develop a novel photosynthetic ethylene production technology using cyanobacteria. This technology has potential to produce both biofuels and green chemicals. And we want to develop a technology that would produce fuels and chemicals as a sustainable alternative to a fossil-based feedstock, and with low water, CO2, and nutrient input, while not competing with agriculture for arable land and fresh water. So the overall scheme of the technology is shown here. I divide that into 3 stages. One is strain development, and next one is reactor development, followed by harvesting and upgrading. So most of the work that has been done so far is in strain development. We have started to work on reactor development. So you can see that there's a lot of work that needs to be done before the technology becomes mature. Next. So why are we working with cyanobacteria? Cyanobacteria are also known as blue-green algae. They are bacteria that perform oxygenic photosynthesis, converting carbon dioxide to organic compounds. They are primary solar energy converters in diverse ecosystems. They are very robust. And for some strains of cyanobacteria, we have excellent genetic engineering tools that allows us to modify its metabolism to introduce new pathways, produce useful products, such as ethylene, and that allow us to further improve metabolic pathways to make more ethylene. Next. So why ethylene? Well, ethylene is one of the most produced organic compounds worldwide. It is the feedstock for a wide range of materials and chemicals that include all kinds of plastics. Ethylene can be polymerized into liquid transportation fuels. And currently, at industrial scale, ethylene is produced from fossil resources. So I want to point out that the fossil resources are organic compounds that are derived from photosynthesis that occurred millions of years ago. So more renewable sources of ethylene are desirable. Next. Here we look at different ethylene production scenarios. So on the left is the conventional fossil resource-based ethylene production. In the middle is a newer plant bioethanol-based approach. And on the right is what we're working on, cyanobacteria bioethylene. So in the conventional ethylene production approach, here we show that we start with petroleum. And that's long-chain hydrocarbons. And petroleum can be steam cracked, processed to produce a mixture of short-chain hydrocarbons. And by cryogenic distillation, you will get ethylene and other chemicals. So in plant bioethanol approach, first you grow a plant, and that's a CO2 fixing process. So you get plant biomass, and then you process the biomass, and you get ethanol by fermentation. Then by a catalytic dehydration process, you can get ethylene. On the right is a cyanobacteria bioethylene scheme. What's shown here is an artist rendition of a photobioreactor that has cyanobacteria in it. The cyanobacteria would accept solar energy and fix carbon dioxide and produce ethylene and oxygen. So in this process, the absorption of carbon dioxide and the production of ethylene happens simultaneously. Next, please. So coming back to the point that all the carbons in ethylene ultimately come from photosynthesis. So in conventional ethylene production process, the photosynthesis happened millions of years ago, and then the biomass was turned into fossils. And now we dig it out, we process it. So what we are getting is the carbon that was fixed by photosynthesis millions of years ago. In the middle, in the plant bioethanol approach, you grow plants, they absorb CO2, and then you process the biomass, you get ethanol, and then convert that into ethylene. So this takes at least one growth season, that will be months. And in cyanobacteria bioethylene production scheme, CO2 is directly converted by cyanobacteria to ethylene. So this is a real-time carbon conversion process. Next. Now, what normally happens with the cyanobacteria, in this case, I work with the strain called Cyanobacteria 6803. So what normally does with this wild-type organism is that it does photosynthesis. So it takes solar energy, it absorbs CO2, it uses water. And photosynthesis produces oxygen that comes out, and the organism makes more cells. Next. To make the organism producing ethylene requires introduction of a single enzyme called EFE, or ethylene forming enzyme. So now, where does this ethylene forming enzyme come from? Next. Now we need to look at biological ethylene production. Ethylene is a well-known plant hormone that regulates many processes in plant growth, as shown here in panel A, that's ripening of tomato. And plant pathogens or symbiotic microbes use ethylene to weaken plant defense, either to establish pathogenesis or establish a symbiotic relationship. So that's shown in panel B. And some of those microbes have ethylene-forming enzyme. The enzyme that we use is sourced from a bacterium, Pseudomonas syringae, that's shown in panel C. So this enzyme is not well understood, which is a challenge for its biotech application. So we know that it takes alpha-ketoglutarate, as shown in panel D, and it requires oxygen. It takes arginine, that makes ethylene, that makes carbon dioxide, and some other compounds. So the reaction is not really well understood. There's no protein structure available. How this reaction happens is still under study. So some of the background is summarized in a review that we published last year, or more than a year ago, at Biotechnology for Biofuels. Next. Now, before we got serious about making ethylene using cyanobacteria, we want to make sure that this product will not be toxic to the organism. So we did some tests. So we added ethylene, and it didn't inhibit any growth. And then we started to generate ethylene producing streams. So they make ethylene and the growth was not slowed down. So we conclude that this product is not toxic to the organism. So that's great news. As you can see on the table on the upper right corner, that we have different strains of wild type and ethylene producing strains. And ethylene producing strains make different amounts of ethylene because they have different copies of number of genes. So one time EFE has one copy of ethylene forming enzyme gene in the genome, and 2 times EFE has 2 copies, and that makes more ethylene. But it doesn't matter how much ethylene they make, they grow as fast as wild type. And we'll get back to that point later. It's very interesting. So now we can make some ethylene. Now can we make more ethylene? So the question is, what is the limiting factor? So a lot of this is published, and the 2 papers. The first paper we published in 2012, that's shown here, in Energy and Environmental Science. Next. So to understand how ethylene is made in a cyanobacteria, we need to look at this scheme shown on the left. So this is a cyanobacteria that does photosynthesis. It takes in carbon dioxide, then it enters metabolic network. And a key component in this is the TCA cycle. So in TCA cycle, there is alpha-ketoglutarate, and which is also the precursor for arginine. And ethylene-forming enzyme takes alpha-ketoglutarate and takes arginine to make ethylene to make succinate. And succinate actually comes back to TCA cycle. And that is also very interesting. I'll get back to that a little bit later. But initially, the level of ethylene-forming enzyme was the limiting factor. So what's shown on the right is what we did in the first 4 years. That is basically to increase the level of ethylene-forming enzyme. So we redesigned the ethylene-forming enzyme gene, and we tested different promoters, and we inserted multiple copies into the genome, and we changed ribosome binding sites. So we used some synthetic ribosome binding sites to increase translation efficiency. Some of those approaches are combined. So in the end, we were able to enhance ethylene-forming enzyme levels. Next. So some data are shown here. On the left of the panel A is a protein gel, shows that between wild type and 4 different ethylene producing strains, we were able to increase the amount of EFE protein steadily. And as a result, as shown in panel B, the ethylene productivity has increased accordingly, and the portion of carbon that becomes ethylene has increased accordingly as well. So in the best strain that was studied in this published paper, this was last year, we published in Nature Plants, the carbon partition rate was up to 10%. So when we measure ethylene productivity, see there are 2 flasks there, and there are lots of bubbles. So in the bubbles contains oxygen and ethylene. And when we do the measurement, we would transfer a small volume of the liquid to a sealed tube and incubate for some time. Then we measure ethylene and CO2 from the headspace using gas chromatography. Next. So that experiment was done in the PG11 synthetic medium. And so here we try to test whether we can grow algae and produce ethylene using seawater, because that's going to be a huge advantage. And this data was published as well. The conclusion is seawater can support the growth of the organism and ethylene production if we supplement that with nitrogen and phosphorus nutrients. And those nutrients happen to be rich in wastewater. So it may be possible in the future to use seawater for ethylene production and recycle the nitrogen and the phosphorus from wastewater. Next. So next question is, well, the organism can make ethylene, but is the system stable? Can you continuously produce ethylene? So to answer that question, we started to work on photobioreactors. And this is one of the early configurations. So we have a 2-liter photobioreactors. And so we have controls of light, control temperature, control pH. There's a stirring mechanism inside to control the stir rate. And we have CO2 feeding in, and we have ethylene and oxygen coming out. Next. So some of the initial data are plotted here. Panel A shows the growth of the culture. And this experiment lasted more than 2 weeks. So the longest continuous growth experiment lasted more than 70 days. So the system is robust and stable. So data from photosynthesis and from ethylene production are shown in panel B and C, respectively. And the ratio of CO2 to ethylene conversion is calculated and shown in panel D. So as you can see here, that in general we get carbon conversion ratio between 2 and 3%. And that is not great. So there's a significant room for improvement. Next. So we took a deeper look into metabolism, and we tried to understand how ethylene production incorporates into photosynthesis, and how can we improve it. But then we found quite a few surprises. So that was published last year in Nature Plants. For details, you can go to that paper, but some of the conclusions are here. In a wild-type cyanobacteria, the TCA cycle doesn't really operate as a cycle. And in ethylene production strain, now they do, the TCA cycle does operate as a cycle. And the carbon flux into TCA cycle increased 3-fold. So the organism is making a lot of ethylene, and it's also losing CO2 by ethylene-forming enzyme. But what was surprising was that the growth rate was maintained. So although the organism is losing a lot of carbon, it grows as fast as wild type. And then we took a look at photosynthesis. We found that photosynthesis is stimulated. So the organism is now taking in more CO2 to compensate for loss by ethylene-forming enzyme. So that's very interesting. There must be an organism, there must be a mechanism that regulates the rate of photosynthesis. And that mechanism is now altered in the ethylene production strains. Next. So some of the data are shown here on photosynthesis. What's shown on the left is the light reaction parameters, including oxygen evolution and the chlorophyll content. And both are increased in the ethylene producing strain. What's shown on the right side is the carbon metabolism parameters. And we saw that the rubisco activity is increased. The carbon partitioning into TCA cycle is increased by 3-fold. And the CO2 fixation rate is also increased. Next. So some conclusions from the work we have done so far is that a cyanobacterium has been genetically engineered to convert carbon dioxide to ethylene. And so far, up to 10% of fixed carbons are directed to ethylene. And the initial rate limiting factor was ethylene-forming enzyme itself. And more recently, as we improved ethylene-forming enzyme activity, the substrate supply has become the rate-limiting factor. So in parallel to this research, we have done some ethylene production study in E. coli, and we have recently published a paper in Biotechnology for Biofuels that shows the strategy we use to overcome this limitation. So the cyanobacteria responds to ethylene production and loss of carbon by EFE enzyme by rewiring metabolic network and stimulating photosynthesis. And that's quite interesting. Next. So clearly we have a lot of work to do. And on the strain development side, we would like to increase carbon conversion efficiency from up to 10% towards 90% using synthetic biology approaches. The rate-limiting factor is now the substrate supply, so we are learning from our E. coli work to genetically engineer the substrate supply pathways. On the reactor development side, we have just started to work on it. And we'll develop cultivation, production, and harvesting system tailored for ethylene production. We're going to need some help there, either academic collaborators or industry. We've started to work on techno-economic analysis, and we plan to work on life cycle analysis in the future. Next. So I want to acknowledge the team. Most of the work was done by talented postdocs, Justin Angara, Wei Xiong, and Bo Wang. And we have a team member listed here, and got a lot of help from NREL photobiology group. So major funding support is from DOE Beetle algae program. And additional support comes from DOE Fuel Cell Technology Office and NREL LDRD. Thank you.
All right, thank you so much, Jianping. That was a fantastic presentation. Our next speaker now is going to be Doug Elliott. Doug Elliott has over 41 years of research and project management experience at Pacific Northwest National Laboratory, with a focus on development of fuels and chemicals from biomass waste. His research has involved biomass liquefaction and hydroprocessing of product oils and catalytic hydrothermal gasification of wet biomass. Within IEA Bioenergy, he serves as the international team leader for the U.S. and the task leader for pyrolysis. I would also like to take a second to extend a special congratulations to Doug, as he is retiring from PNNL this week. And Doug, it's all yours.
Thank you. Thank you for the introduction. So today I would like to talk about the hydrothermal process that's being developed at Pacific Northwest National Lab. We were very pleased when we earlier found that we had been recognized as one of the finalists for the R&D 100 award, and even more excited when we actually won the award at the presentation back in November. It is a team effort. I'm just the spokesman for this process work this morning. Next slide. So just as an overview of what we're after to present to you today is a process that converts wet biomass material into fuel products. It is both liquefaction to produce liquid fuels, but it is also a gasification technology to produce some extra fuel gas product that can be reused as part of the liquefaction or could be reused as fuel. Important elements to recognize about the work that we are doing in this field are that we are working in continuous processing systems. Our technology is at the stage where we can design it for scale up and build it into bigger plants. and we see it moving forward from our laboratory in rather short order. We have been working with a whole range of wet biomass materials that cross the whole spectrum from lignocellulosic to algae, including various wet waste materials and even wastewater treatment sludges. And a key part of what we do is, in the continuous flow system, produce an oil product that spontaneously separates from the byproduct water phase, and we allow for mineral separation as a solid byproduct so that our technology can be applied to this vast array of feedstocks known as biomass and can handle the range of compositions that are found in those kinds of feedstocks. I'll also talk a little bit about the upgrading that we do of the bio-crude product from the liquefaction step. It's catalytic hydrotreating to clean up the sweet crude really that's being produced in our case and make a low oxygen hydrocarbon material. I'll talk about the CHG, the catalytic hydrothermal gasification of the aqueous phase to clean it up and allow for recovery of the nutrients as well as the energy value of the organic material in the water. And then we'll finish with where we're going with this technology into commercialization. Next slide, please. This technology isn't brand new. It has been under development in this laboratory off and on since the beginning of my career here. So there was actually a plant built in the 1970s after the first oil crises that was operated into the early 1980s, and it's pictured here. This technology was analyzed and passed over in favor of fast pyrolysis because it was really looked at only from the point of view of making a fuel oil product. And you can see from the flow chart here, it's rather complex. To work with wood actually required an acid hydrolysis to break the wood down so that it could be pumped as a slurry and try to generate higher concentrations of biomass in water. Unfortunately, the liquefaction step is done more at a higher pH, so alkali addition was required then to neutralize the acid as well as provide the catalyst, which functioned in combination with the syngas, the carbon monoxide hydrogen that you see being added, and a kind of complex chemistry based primarily on formate reduction to convert the oxygenated biopolymers into the bio-crude product. But as you can see, it goes then on through the high temperature, high pressure tubular systems, cools down, separate out the water, a significant amount of dirty water, and produce a crude product. So it's basically what we would say was pyrolysis, a slow pyrolysis in a pH moderated system functioning in pressurized liquid water. So the next click. So we had to make some changes. This technology wasn't competitive with fast pyrolysis, and we do it differently today in order to make it competitive, and in fact we think it may be even a more valuable technology than pyrolysis for making finished hydrocarbon fuels. First of all, we do not any longer convert a significant part of the feedstock into the reducing gas. No gasifiers involved, there's less capital equipment, there's no diversion of carbon into that kind of reducing gas and gas products that drive the system. The other thing that we don't do anymore, next click, is we don't worry about trying to break down the biomass with acid hydrolysis and having to subsequently neutralize it. That step is completely eliminated. We just do a grinding, wet grinding, dry grinding to make a slurry. we're only able to achieve concentrations about 13% that are still pumpable in high pressure pumps. But our analysis, the techno-economic analysis that has been done on these operations suggests that this is still a reasonable and more economic way to proceed. But the key difference is, next click, we don't think of it as making heavy fuel oil anymore. We think of it as making a crude product, crude oil. We look at the upgrading of that to make finished hydrocarbon fuels, diesel, gasoline, jet fuel. And in that processing step, the liquefaction has great advantages over fast pyrolysis. Our biocrude product from HTL is much more easily processed. It requires less hydrogen to get it to the finished products. And this is a very significant difference in how we look at this technology and how we use it today compared to back in the 80s when this plant pictured was shut down and the technology was basically abandoned. Next slide. So what we are talking about is processing at high temperature and pressure, about 350 degrees Celsius, just under the critical point of water. So it is still a liquid water moderated environment. It requires high pressure to maintain that environment. So pumping biomass into such a high pressure system requires the biomass be in very small particle sizes into slurries, as shown in the pictures here of wood and straws that can then be processed in the high temperature environment to make it into a crude oil. There is this significant dirty water aqueous byproduct that comes out. There is carbon that goes in that direction. We do need to recover that to make a economical process overall. But we've shown now that we can take that biocrude, we can hydrotreat it through catalytic processing that is not all that different from petroleum, well-known petroleum processes, and make finished hydrocarbon products that can be distilled, as you see in the lower left side, the light gasoline phase, a jet diesel phase, and then heavier oil products. Next slide. As I mentioned, there are these range of products. There is an oil product. It is phase separated from the water, so it is the water insoluble portion that we work with and further hydrotreat. We don't do any kind of solvent extraction to recover, as is commonly done in small batch reactor tests that are reported in the literature. It is a more viscous oil than is produced in fast pyrolysis, but it still is fluid and will work, can be moved and pumped through further processing systems. It's a reasonable yield on a mass basis, varies depending on the particular biomass feedstock, anywhere from 25 to 50% on a mass basis, but more importantly on a carbon basis, it can be as much as 70%, and you'll see in some of the algae tests we've shown even up to 80% of the carbon ends up in the biocrude, this water separable biocrude product. Now, there also is still a small amount of water dissolved in these products because they are still partially oxygenated. They've been greatly deoxygenated compared to just a simple pyrolysis process. We do produce a little bit of gas byproduct. This is primarily a deoxygenation step because the product is essentially carbon dioxide. It's not a major loss, but it certainly is a very significant step to removing oxygen from the biomass and getting us on the pathway toward hydrocarbon fuels. We do have aqueous phase that is produced, a large amount of it in fact, and it does contain a small amount of carbon, typically in the range of about 2% carbon, and these are primarily the organic acids and light oxygenates. We're more than happy to let them go out into the aqueous phase because these components would not be good feedstock for the hydrotreater, would not produce liquid hydrocarbon products, but in fact through hydrogenation, through hydrotreating, would only produce more light gas products. So since we are trying to maximize the liquid hydrocarbon product, we're happy to let that carbon go out into the aqueous and process it separately. And or a very important aspect of our process is we have engineered and developed systems that allow processing and separating of mineral content in biomass that can be stripped out of the system at reaction conditions. This isn't a subsequent processing step, but actually happens online in the system. We precipitate primarily the calcium magnesium phosphates, but also some sulfates come out in this range, in this product. So this is a key way to recover, separate phosphorus and allow it to be recycled as a nutrient. There is a small amount of carbon that goes out in that product as well, and would of course then be lost from the good oil product that we're trying to yield. Next slide. This design laid out here is our bench scale systems at Pacific Northwest. We've done very extensive work, made several publications over the last years, designing, rebuilding, refining the reactor systems. As shown here, there are a couple stages of pumping, low pressure pumps, higher pressure pumps, potential for tubular preheaters, potential for a continuous stirred tank reactor, and even plug flow tubular reactors for high temperature. Our focus of the research these days is to maintain a strictly plug flow operation. We found we can do that now by maintaining velocities at sufficient levels, and have taken really the stirred tank reactor out of the system. Engineering sense says to get rid of that unit. Tubular reactors are much easier to design, run, and operate, much lower maintenance from a processing point of view, much more reliable in the long run. But it is a relatively simple process in that we're basically pumping to high pressure, heating to temperature, letting the chemistry occur over some minutes, and then cooling it, separating it and cooling back down. Here you see the solid separation step. The liquid collectors then, once the mineral solids are separated, those collectors work very effectively for phase separation without emulsion formation, a key difference from other research out in the literature and other process technologies out there. And then finally we recover the gas products and analyze those to complete our mass balances. Next slide. So I will give you just a hint of the different kinds of feedstocks and results we get. We have more publications out there if you care to follow up with those and get these details, but as I understand, these slides at least will be up after the seminar is over, and you can review those at your leisure. But you can see here we've worked with a range of lignocellulosics, all the way from pure cellulose through grass materials, even to wood. These can be made into pumpable slurries, ranging from 13 to 15% dry solids basis, and can be pumped and processed in our system. Next slide. The interesting thing is that converting of cellulose works almost as well as the other lignocellulosics, but in fact the lignin does seem to give a higher oil yield. The cellulose, of course, has almost no mineral and produces almost no solid byproduct, has a higher yield into the aqueous phase. But this whole range of wheat straw, switchgrass, and wood, you can see that the oil yields are similar in all cases. The product distribution is similar for these. Processing at a range here in this table shown as about 2 liters an hour. In fact, we're finding that we can push this even faster through our systems. Most recent results are showing 2 to 3 times the increase in the space velocity through the reactor with a purely plug flow system. Next slide. Here is the breakdown then of comparing, whereas we reported mass yield on the previous slide, here it is on a carbon basis. And you can see that clearly the oil yield is the primary product we're making when you look at it as a carbon basis. We think this is a good yield that can be economically viable in this range, 45 to 60% yield of a biocrude product. The other major product and the other major fate of the carbon is into the water stream, shown in black. A little bit of gas, a little bit of char. Good balances on most of these experiments. These are for operations that last about a day, a long day, and we work for steady state operation on our continuous flow system and take operating windows and collect data that are from windows 2 to 3 hours long. Next slide, please. The composition then of the crude product that we're making, as you can see in this table of data, are fairly similar depending on the feedstock. There are some minor differences. Very interesting point to consider is that cellulose makes a highly aromatic oil by hydrothermal liquefaction, something that is not widely recognized. The oil does not just come from lignin, it comes from the cellulose as well, and as you'll see later, the oil also comes from protein and other biopolymers. But just even pure cellulose produces a product oil that has a hydrogen to carbon ratio of just over 1. This is a very highly aromatic phenolic material. The oxygen contents are much lower than in pyrolytic pyrolysis oil products. You can see here ranging around plus or minus 5 or 6% from 10. Typically about 10%. Almost no nitrogen, almost no sulfur, because those feedstocks have very little of those. But where they are present in the wheat straw and the switchgrass, for example, you see them coming through in the product. It is a much lower TAN compared to a pyrolysis oil. Of course, this is much higher than petroleum because there is this very significant oxygen content still there. It is a heavy oil. It is actually heavier than water. The density is here about 1.1. Viscosity is, as I mentioned earlier, viscous material, but fairly clean oils, low ash, low solids contents, but still significant water dissolved in it. But compared to a pyrolysis oil, which can have in the range of 25 or 30% water, this is a more condensed, a more energy-dense oil than from pyrolysis oil. Next. We've also applied the technology to algae, and in this case, we find it particularly interesting to show how algae can be grown, and it needs to be concentrated. The item number 1 there, a dewatering system, recycle much of the growth environment back to the algae growing system to collect the solids. We don't need to dry it though in our system. We're still working with wet algae, typically around 20% dry solids of wet algae. That can be accomplished with mechanical dewatering as opposed to thermal, so the energy input is relatively low at that point. We do the liquefaction in step 2, and you can see in step 3 where we separate water and oil. The water carries nutrients and solids, also separate out and can be used to recycle back for more algae growth. The liquids go on to 4 where the oil and water separate. The catalyst is of course different in steps 5 and 6. It's a hydrotreating catalyst to convert the biocrude into the finished hydrocarbons. It's our special aqueous phase catalyst for gasifying the organics in the water stream to produce a methane CO2 product gas. Next slide. We can make this liquefaction on both whole algae, or as we started in the early days, the lipid extracted algae. If one wanted to recover the hydrocarbon separately, we can still make oil out of the lipid extracted algae material, biomass. If there are other technologies, which we are also working on, where there are other products to recover from the algae and then do the liquefaction on the leftovers, this clearly is an option. But it makes a hydrocarbon product as shown in the chromatograph at the bottom, which is a whole collection of materials from the cyclics generated more from the protein and the carbohydrates, and then the long chain hydrocarbons left over from the lipids. Next slide. And to show, to underline this aspect, we actually did work with 2 strains of algae, one low in lipid, one high in lipid, same algae but just grown under different conditions. We showed that this product of biocrude could be produced from either of these, whether there was a lot of lipid or not. Good oil yields, as you can see here, the high carbon yield of 80%, and making a biocrude not unlike the lignocellulosic in that it still has significant oxygen in it, but less than 10% in the algae case, but still the higher protein content in algae versus lignocellulosic shows up here, 4% nitrogen in the biocrude and a bit of sulfur. Of course, the hydrotreating, the upgrading, is where these final bits of oxygen, nitrogen, and sulfur are removed to produce a pure hydrocarbon fuel. Next slide. We do the hydrotreating at Pacific Northwest Lab. We have several scales of operation that we can perform this kind of process. This shows what we call our mini reactor systems, but it is a simple tubular reactor. We have shown that it can be done with a single stage of processing, unlike a pyrolysis, a less stable pyrolysis oil. This is an important aspect to making this a more cost-effective hydrocarbon product coming out. But again, removing oxygen, sulfur, nitrogen, a little bit of hydrocracking to break down molecular weight, and some isomerization may be needed depending on the fate, the market for the finished fuels. Typically running at about 400 degrees C and 1,500 pounds per square inch. Not terribly different from what a petroleum refinery operates at. We do full mass balance on these kinds of systems, so we know how much hydrogen is being consumed. We don't have to just base it on calculations and assumptions. We actually produce the data which allow for design of these systems at larger scale. And we're using conventional hydrotreating catalysts. These are sulfided molybdenum catalysts, cobalt or nickel promoted. We prefer cobalt promoting for most of our work. Next slide. With this kind of processing, then you can see that we make a good product shown in the table on the right, the upgraded HT product, low in oxygen, nitrogen, and sulfur, removed down to a very low level, only 7 parts per million in this particular product, having the properties one would expect of such a hydrocarbon: a low density, low viscosity, low water solubility, low acid content. What this slide also shows, though, on the left-hand side, the simulated distillation charts for several products produced over time in our operating system. Quality controlled diesel in blue, but then the red, green, and purple are time on stream, 34 hours, 118 hours, even up to 208 hours, and you see that the 3 products are almost identical. Very difficult to see any significant catalyst deactivation under this operating environment. This, of course, is not sufficient for a refinery. They need to run for months and years online without significant changes. But this is a very good first step, and certainly more than you'll find in the literature, I think, from any other research group. Next slide. These same kinds of products can be made by hydrotreating the algae biocrude. You can see density data, all the same kinds of data. even with the higher nitrogen content in those algae-derived biocrudes, nitrogen is down to a very low level. It is not, in the case of this kind of biocrude, the nitrogen components are not the killer, not the scary topic that the petroleum people are concerned about, because our nitrogen is typically in the form of simply hydrotreated products like amides on the fatty acids and simple heterocyclics not buried in porphyrin structures that are a very complex polycyclic typically found in petroleum. Next slide. So these, just to show that these products can be distilled and used in the range of typical fuels, here it shows some data where we just mixed all of the products together. They can be distilled into naphtha, jet fuel, diesel. Next slide, please. The products are analyzed then, and we have the data to show that these are the quality of material that could be of interest to petroleum refiners. Next slide. And just finally, again, very detailed analyses are required to get to quality of diesel and jet fuels. Very low sulfur levels are required. The cloud point, pour point, freezing point data are key for operation with jet fuel, and these products have the right properties to be used in that fraction relatively straight away from the production out of our hydrotreating. Next slide. The gasification I mentioned, it is a way to clean it up. The picture shows how algae slurry gets to a biocrude and a dirty water, and we literally can make that kind of clear white water out of the product. It is a catalytic process. It is we are having some issues still cleaning up the water well enough that we can process over a catalyst, and that's a subject of more research. Next slide. We're very pleased to be working with Genafuel. These are the folks that have taken our technology, scaled it up, built the plant as shown here. It's on its way to its customer site for further testing and development. With Genafuel, we did receive the Federal Laboratory Consortium Award for Excellence in Technology Transfer last year. And then, of course, they are our co-winners on the R&D 100 award. Next. Just showing that we hope to be making it at making the finished products at larger scale. We have this scaled up hydrotreater, which we hope to be running later this year to make larger quantities, gallon quantities of fuels for testing. Next. So we've worked with all range of wet biomass materials as shown here, from food waste to algae. We are building a larger scale version for further testing. Our economics, we think, have been studied and shown to be of interest. We're working to larger scale operations. Next. So we have a continuous flow system. We make quality oil straight away. We can hydrotreat it to make finished products. We're looking at a whole range of feeds. So we think this is a very important development. We're pleased to be recognized. Next slide. And as I said, there's a whole team of people at the lab. The people that were recognized on the award, but other operators in the liquefaction, the hydrotreating, the analytical people, the techno-economic assessment people, and life cycle analysis. All of these folks at our laboratory have contributed to where we are at on this and receiving this award, and appreciate all of their efforts. And thank you.
Thanks so much, Doug. With that concludes our webinar this afternoon. You can see how these two great projects and these two great teams are helping to support the development of sustainable, renewable energy alternatives, and we are very proud to have them as part of the BETO family. If you've submitted a question to us today, we have it, and we will get your questions to our speakers today. And if you have any questions after this webinar, please again email this address here, EERE_bio bioenergy@ee.doe.gov and include R&D 100 webinar in the subject line so we know what you're talking about. I'll keep this on a little bit longer so you can submit any questions here, but again, please submit them via email otherwise. And another big thank you to our speakers today, Doug and Jianping, and to everyone else, have a great afternoon.